First Light. Antimatter Society. Episode 4. The 43rd batch died on the 9th day. Naya made the entry in her research log the same way she made all the others: date, batch number, conditions, time of death, preliminary observations. She had been keeping the log in this format since the project began. Spare, precise, no editorializing. The facts were the record. Her interpretations were a separate document, and that document was considerably more uncertain than the log. She had grown organisms from synthetic genomes before. She had worked from reconstructed sequences, degraded samples, theoretical designs. She knew how to build life from a description. She had been doing it for most of her career. This was the first time what she built would eventually ask where it came from. The genome she had received from the fold station, transmitted from the antimatter side, relayed through the station at the fold mouth, arrived as a data package the size of her team's combined library, was not a degraded sample or a theoretical design. It was a complete and precise specification of a person. It was, as far as she could tell, the most detailed biological blueprint she had ever worked from. And everything she grew from it died. Not immediately. Not in any way she could point to as a clean failure mode. The cells developed initially, showed expected responses, appeared viable through the early stages. Then, around the 7th or 8th day, something began to go wrong at a level her instruments could detect but not characterize. By the 9th day, they were gone. She had eliminated technique. She had eliminated environment. She had eliminated contamination. She had run the diagnostic sequence 4 times and found nothing she could act on. What remained was the source material, something in the genome that her standard processes were handling incorrectly in a way she had not yet been able to name. She added a note to her uncertainty document: source material incompatibility, mechanism unknown. The standard processes were the point. Growing an organism from a synthetic genome was solved science. Her team had done it hundreds of times with matter universe genomes, native sequences, well-characterized biology. The equipment was correct. The protocols were correct. Everything was correct, and the cells kept dying, which meant the problem was in the one thing that was not like anything she had worked with before, a genome that had not evolved in this universe. She thought, as she sometimes did when the instruments were running their overnight cycles, about the scale of what she was part of: the fold station, the colony team, people working on aspects of the project she had no visibility into. She was one piece of it. That was sufficient. She closed the log and began preparing the 44th batch. She ate at her desk more often than she should have. That evening, it was bread and something preserved, the kind of meal that required no attention. She had been thinking about the 44th batch, which had reached day 7 and was showing the same early-stage viability that always preceded failure. She was not ready to go home. She bit into the bread without registering the taste. Then she registered it. Then she stopped. Taste receptors worked chirally. She had known this for years. The molecular shape of a flavor compound mattered as much as its composition. Mirror image molecules could taste completely different from each other. L amino acids and D amino acids had the same atoms in different orientations, and the tongue knew the difference, had always known the difference, because the whole tasting apparatus had calibrated itself over millions of years to one particular handedness of molecule. Her tongue worked correctly because her biology and her food had co-evolved in the same universe, using the same chirality. The antimatter civilization had evolved in a different universe. She set down the bread. In the matter universe, life had settled on L amino acids, the left-handed version. The reason was probably historical accident. Whichever chirality had gained an early foothold had locked in, and everything downstream had built on that foundation. In an antimatter universe, the same process would have run, but there was no particular reason it would have gone the same way. CP violation. The fundamental asymmetry between matter and antimatter that determines which way molecules spin meant that a different physical history might have produced a different outcome. Life might have settled on D-amino acids instead. The same logic, the opposite outcome. And she had been building from their genome using standard matter, universe molecular machinery, all amino acids, the chirality her protocols were built for, producing cells that were internally inconsistent. Some molecular components in one orientation, some in the other. Viable through the early stages, then failing as the inconsistency accumulated, as the proteins that should have fitted together found that they didn't quite, because they had been built from components that pointed in incompatible directions. The fix was to build the mirror. She left the bread on her desk and went back to the lab. The 45th batch used D-amino acids throughout, mirror image sugars in the nucleic acids, every molecular component inverted from her standard protocol. She worked carefully and without hurrying, checking each step. She had the feeling she sometimes got when a problem she had been approaching from the wrong direction finally revealed its correct angle. Not excitement exactly, but a focused clarity, the sensation of asking the right question for the first time after a long run of wrong ones. On the 9th day, the cells were alive. On the 10th day, they were still alive. She sat in front of the culture chamber for a long time. The readout was unambiguous. The cells were viable, mirror image cells built from the inverted specification, every molecule the opposite hand from what the original antimatter organism had been. The blueprint had been followed exactly. The building was the mirror of the original. The building was real. She wrote one line in the research log, batch 45, day 10, viable. She did not write anything else. The rest would take longer to say. The documentation took 3 days. She began with the chirality solution, the mechanism, the reasoning, the specific inversions required. This section was precise and complete. She expected other scientists to build on it, and she wrote it so they could. She noted what the mirror image construction meant for the resulting biology. The organism would be internally consistent and viable, but its molecules would be the opposite hand from standard matter universe life. This had consequences that she was obliged to name. Taste and smell. The flavor receptors and olfactory system would be mirror image relative to the molecular landscape of the matter universe. Food would be processed correctly. Digestion was a function of chemistry, and the chemistry would work, but the experience of tasting it would be systematically off. Not wrong, not unpleasant, just not what the genome had been calibrated to expect. She could not fix this. It was a consequence of the only viable construction method. Vision. The visual pigments in the genome were optimized for the light output of the antimatter homeworld star. The matter universe planet chosen for the colony had a different sun, with a slightly different spectral profile. The mirror image construction compounded this. The visual system would receive light from a sun it had not been built for, processed by receptors in the wrong chirality for the matter, universe photon interactions. Colors would be subtly and persistently off. She documented the predicted deviation in detail. She could not fix this either. The star was the star. Protein interactions. The mirror image proteins would fold correctly relative to each other but would interact with matter, universe enzymes and environmental proteins in subtly different ways. Most metabolic functions would be unaffected. Some would not. She listed the affected pathways and her best estimates of the magnitude of deviation. She noted that these were estimates. The biology of a living organism was more complex than the models, and the real deviations might differ from her predictions in ways she could not anticipate. The microbiome. The antimatter civilization had co-evolved alongside antimatter microorganisms. No ancestral microbiome existed in the matter universe. The colony team would introduce a synthesized one. She had worked with them on its composition. But a synthesized microbiome was not an ancestral one, and the genome had not been built alongside it. She expected the resulting organism to be healthy. She expected the nutrition to be less efficient than optimal, in ways that might not be measurable in any single generation but would accumulate over time. She noted this. She wrote all of it. She wrote it carefully and without apology, in the same spare register she used for the research log, because the record was the record, and the record had to be honest. The people who would eventually read these notes, in the archive perhaps, or in whatever documentation made it to the other side, deserved to know what had been possible and what had not. She owed them that, even if she would never meet them. When the documentation was complete, she assembled the full package, the corrected genome blueprint, the chirality protocols, the supporting experimental record, and the notes she had just written. She transmitted the package to the colony team. They had found the planet. The terraforming was underway. In some number of years, more than she would count eagerly, the colony would be ready, and the blueprint she had just sent would be built into living people. She sat for a while after sending it. She had grown organisms from synthetic genomes before. This was the first time she had grown one that she would not follow. Every other project she had closed by moving to the next phase, the observation period, the developmental studies, the interaction experiments. That was how the work progressed. You made something and you watched what it became. The agreement was clear. The Matter civilization's role was to establish the conditions, find the planet, terraform it, grow the first generation from the blueprint she had developed, support them until they were self-sufficient. After that, withdrawal. Complete and permanent. No ongoing contact, no observation from a distance, no checking whether the predictions in her notes were correct or which of the divergences she had named turned out to matter most in a life actually lived. That was not the agreement. The agreement was that these people would be the antimatter civilization's people, shaped by the antimatter civilization's transmissions and culture, becoming whatever this universe made of them, without the matter civilization's presence distorting the result. She understood the reasoning. She had understood it when she took the project on. The people she had made possible would read her notes, if they found them, and know her as one of the engineers, not by name, not as the person who had sat at a bench until the early hours synthesizing mirror image molecules, who had eaten cold bread at her desk and arrived at the answer through a thought about taste receptors. Just as the engineers, plural, the professional category, the careful and apologetic technical record. That was appropriate. That was what the agreement asked of her. She closed the documentation file and looked at the culture chamber across the lab. Batch 45 was still alive in there. The original cells, the proof of concept, the first light. They would be maintained until the colony team no longer needed the reference sample. Then they would be archived and eventually decommissioned, and the only things that would remain were the notes and the blueprint and the people who would one day be grown from it. She thought about those people tasting food that was slightly wrong, looking at a sky whose colors were not quite what their eyes had been built for, living in a world that fit them almost exactly. She thought about them not knowing that any of this was the case because they would have no reference. The wrongness would simply be the texture of things, the way the world tasted, the way the light fell. She thought this was not a failure. She had done what the physics allowed. She had made them real. She turned off the bench light and left the lab. The blueprint was transmitted. The colony team had what they needed. In some number of years, the first of them would be born into a world that fit them almost exactly, and they would find it sufficient, and she would not be there to see it, and that was the shape of the work she had done. She walked out into the Matter universe night and did not look back.